Hi, great to have you all here. We're starting with some greeting words from Itzik Chen, the director of the Institute. בבקשה.
Thank you, and welcome all to Jerusalem and to the Israel Institute for Advanced Studies. The Institute is celebrating its 50th anniversary this year, and there's nothing more appropriate to open this festive year with the annual Yeast meeting. I won't tell you much about the Institute because you can read it all. We have a very nice exhibition downstairs on the walls. I'll just say that each year the Institute hosts a research group of scholars from all fields. This year we have a group on non-verbal communication in the Islamic world, and a group of mathematicians who are working on spectral theory of graphs. We also have individual fellows, we have an author in residence, and we have some artists that are associated with the Institute. Each year we hold 6 advanced school for PhD students. And in your field, the one that is relevant is on life sciences, which is organized every year by Roger Kornberg. And I do hope to see many of you in the next advanced school. But we also in charge of the Bruno Prize, which is given by the Yad Nadiv. And the reason that you are here is because Maya got the Bruno Prize last year. So I won't say anything interesting about yeasts because I'm not in the field. I'm from the other side of the campus or spectrum, if you want. I'm an historian of the early Middle Ages. So we did have yeast, but we didn't call them yeasts yet. So I'll just wish you a successful and interesting workshop today, and as I should greet yeasty people, rise and shine. Thank you.
Thank you, Itzik. Just a second. Okay. So good to see everyone. After 2 challenging years marked by war and uncertainty, Maya Schuldiner and I felt it is especially appropriate to devote this year to focus on the theme of stress. Yeast and people are overcoming stress with the hope that together we can turn the topic of stress into an opportunity to learn from one another and to strengthen our sense of community. As scientists, we trust evidence, and the research is becoming increasingly clear about this. Strong scientific communities and supporting professional networks play important role in academic success, collaboration, and growth, as the papers I gathered here highlight very clearly. This paper, for example, showed that collaboration networks act as a social capital that helps research thrive. In-person conferences, as the one we hold today, offer unique values for sharing ideas, having real conversation, and sparking new collaborations. Although virtual conferences and meetings are possible, face-to-face interaction really create connections and opportunity that simply are not that easy online. I like this piece of editorial comment where the editor beautifully wrote that scientific conferences provide a valuable support networks, especially during the early stages of scientific career. So we thank you very much for making the effort to come here to Jerusalem, and I think this effort resonates strongly with the message behind the findings that meeting in person truly matters. At this year's meeting, we not only want to get updated, but we want to really help our students. So we've added a special de-stress panel where researchers will share their own journey in academia and will answer questions that you guys have on career path, very important key decisions in your academic avenue. Before we begin, we have one small request. We ask researchers here to consider hosting and organizing next year's meeting in your institute. Throughout the day, Maya and I will walk through and...
I'll do it.
Really? Yeah, Shai is the best. Perfect. Thank you so much. Unless someone else is volunteering. Okay. That was really easy and great. Thank you.
It really sounded planted.
Yes, we are. That's true. And now, it is my true pleasure to introduce our first speaker, Professor Tzachi Pilpel, who will take us from nature to the lab and back again on the topic of yeast adaptation to stress. So please, Tzachi.
You know, I'm interested in mistakes, and my name was spelled with a mistake, T-S-A-C-H. And that, to me, it's an indication, when mistake happens, it's interesting at what level it happened. Obviously, S and Z are not close on the keyboard to one another, but they are close vocally, and that will resonate with some of the mistakes that I'll show you today. Than in Yeast. Really, I mean, the type of keyboard that you're using reveals the type of, or the mistakes reveal the keyboard. I was contemplating what to talk about here today, and Maya and I discussed it several months ago with inputs from Ayelet, of course, too. And we thought maybe, but I take full responsibility on that, maybe rather than focusing on one story from the beginning to the end, I will do something unusual, but Vil says that I always do something a little unusual in my talks, so this is my usual. But it's unusual in a different way because I will take you with me on a journey of many, many years in which we have been using many times yeast, not necessarily because we were interested in yeast for its own sake, but because of its capacity to lead us into understanding principles that prevail perhaps outside of yeast, perhaps throughout biology, or at least in some other cases. So this is where I come from to yeast, and so it's gonna be kind of intense. Many little snippets, each of which is on a totally different project. The only thing that they have in common is our sick mind is interested in all of them. But really, I think what would happen again and again is that you see how a yeast observation led us to something outside of yeast, perhaps of some generality. So let's start. Many years ago, close to 20 years ago, Onan and myself have looked into codon usage of yeast genes and how well the codon usage of a gene adapts to the tRNA pool of the organism. And we found something really interesting. We found that genes that belong to the same pathway, they tend to have the same level of adaptation of their codon usage to the tRNA pool. In particular, if we looked at all the glycolytic enzymes in Saccharomyces cerevisiae, we saw that all of them have a very good codon adaptation to the tRNA pool. But if we looked at the Krebs cycle genes, we found that they invariably have a much less well codon adaptation to the tRNA pool compared to the glycolytic enzymes. We said, okay, yeast, Saccharomyces cerevisiae, it's a fermentative species. Let's see what happens in non-fermentative species that prefer respiration, either obligatorily or facultatively. And then we saw almost exactly the mirror image of that. In fact, we developed the means by bioinformatics to look at the codon usage of certain genes and determine whether a yeast would like to prefer or to ferment, to respire or to ferment, depending on the codon usage of those genes and some other genes. In fact, when we looked at the entire yeast genome, we found that the main axis that spans codon usage adaptation to the tRNA pool is this fermentation respiration axis. And different organisms kind of switch places along this axis depending on the lifestyle. So that was an interesting lesson from yeast. And then we asked, does any of that hold, for example, when we come to mammalian species? And then we found something totally different, but with the same principle in mind. We found that the main axis that separates codon adaptation of genes to the tRNA pool is not fermentation respiration, perhaps not surprising, but rather whether the genes are involved in what we call unicellularity functions or multicellularity functions. You know, every gene in our body is either involved in a cellular function, such as replicating the DNA, building a membrane, or to communicate between the cells in the body, generate a developmental pathway, response to hormones, etc., etc. We found that this main axis of uni and multicellularity is what separates the codon usage programs. And indeed, they are different between the two types of genes, and the tRNA pool itself corresponds to that. When a cell is dividing, it's mainly expressing the tRNAs that serve the codons that are needed by unicellularity genes. And when a cell is differentiated or respond to some multicellularity functions and instructions, it shifts its tRNA pool so as to serve the codon usage of the genes that are needed there. In fact, we discovered that by looking at the cancerous tRNA pool that showed a massive reprogramming of its structure, and that led us to understand that the main axis that spans codon usage programs in mammals, and it was conserved outside even of mammals, was that of uni and multicellularity. So it started from yeast fermentation respiration, and it led us to reveal something interesting about mammals. So we, as mentioned, and it fits the theme of this conference, we love to stress yeast in the lab and then we see how they respond. And in addition to stressing them by a variety of physiological conditions, we also like to stress them genetically too. So we mutate their genome, or we insert a gene that disrupts something, and then we like to see how evolution in the lab allows them to relax that perturbation. So one of the perturbations that we did was again in the tRNA pool. We love tRNAs, we love translation apparatus, etc. So we decided to do a very nasty small change in the yeast tRNA pool to delete one tRNA that appears in a single copy in the cell and let cells evolve towards that. This was a tRNA for arginine from an anticodon that there is only one copy. For many tRNAs, there are multiple copies. For example, there is another tRNA for arginine that has 11 copies. So maybe it can spare one of them, but it has a different anticodon, so it cannot rescue the organism. When we grow the cells in the lab, this is the wild type, the green, this is the mutant, the blue, you see a severe fitness disadvantage. But if we evolve the strain over by serial passaging over just a few hundreds of generations, you get the red curve, which totally closes the gap. So within just a few weeks of evolution, they overcome the fitness deficiency that is conferred upon them when they lose a single copied tRNA gene. And then we wanted to understand how did they do that. Rather than sequencing the whole genome, we started a gamble in the lab. Who would perhaps guess what was the mutation, if it is a mutation, that allowed them to overcome the challenge? I don't know if you have a quick guess, but one of the guesses was maybe something happens in the tRNAs and in the anticodons of the tRNAs that are serving for the same amino acid, albeit with a different anticodon. So we decided just to sequence those tRNAs and see what happens. And indeed, we revealed that the mutation always occurred in one of the other tRNAs that have a different anticodon, and we revealed that in fact what happens is that they go to the genome and change exactly the wrong nucleotide in that anticodon to become identical to the anticodon that would have been in the other tRNA that we deleted. It was interesting and not trivial because those two tRNAs differ in 20 additional nucleotide positions out of 72. So, but it turns out that just changing that was sufficient. So that was a whole story. It's published. The references appear here below. It was a whole story. I'm not going you through that story, and beyond just saying that it then gave us, and this is indeed the notion of this lecture of going back and forth from yeast to outside of yeast and from lab to nature. We said if it happened, this anti-codon switching happened in our case so quickly in the lab, maybe it happens in nature too. And we devised a whole series of algorithms to reveal cases where the tRNA pool switches between different tRNAs by merely mutating anticodons. And we found that this is a very pervasive and prevalent mode of evolution of the tRNA pools. So you have the tRNA pools, they are very different in their sequence, but kind of a network, a mutational network connects between tRNAs by converting them between one anticodon into another, sometimes within the boundary of the same amino acid, and interestingly, even outside the boundaries of the same amino acid, revealing a very dynamic nature of mutational events that can move the tRNA pool according to demand. Okay. Another totally unrelated story done with Martin Kupiak here was that we published again many years ago. And again, the only thing it has in common is that we did that, and it stresses us and yeast as well. We wanted to understand, it was all based on Amir Mitchell's idea back then, my student and our professor at the University of Massachusetts. He came to me at the beginning of his PhD and said, Tzachi, we know so much about how yeast responds to the current condition in their environment. But do they have any capacity to predict a future change and to prepare to head ahead of time? And trivial response would be, well, animals, humans can do that. We have brains. With a brain we can predict and prepare and anticipate, but we don't know that unicellular such as yeast have that capacity. But we said, we have a teaching from psychology. Pavlov taught us how dogs, for example, can condition between one stimulus and into another, the ring of a bell and a piece of meat in that case. Is there other, in the physiology and ecology of yeast and bacteria, pairs of bells and meats, such that through evolution, it's not through a physiological process that happens within the brain or the cell of a single individual, but throughout evolution perhaps generate a possibility to do Pavlovian-like conditioning. So we focused on yeast and in E. coli. Obviously I will focus here today on yeast. And a prime ecological situation that applies to yeast is the process of wine production. We learned about wine production and the series of ecological changes that occur in the barrels during the wine production process. And without going into any of the details, I would just say that this is a very stressful process. Many things happen. At certain pivotal point in time, there is a dioxic shift because they run out of glucose and they start to respire rather than to ferment. And the question was whether any of those stages serves as a Pavlovian bell that prepares the yeast for a meat. Although it doesn't have to be a good thing necessarily, it could be a bad thing that you might want to prepare to perhaps even more vigorously, that would come later on. And since yeast have evolved for many, many years, and many of them remain at the bottom of the barrels of the fermentation process, if a mutant arises that can predict ahead of time what would be the next stress based on an original one and a previous one, perhaps that mutant may have an advantage, and we'll see that yeast have that capacity. I'm not taking you through any of that, but together with Martin, we have verified that indeed Saccharomyces cerevisiae has an ability to predict and to prepare ahead of time. If you prime it with one of the initial pressures, it is coping better with some of the later ones, but it doesn't work if you do it in the opposite direction, in the opposite temporal direction. And we even found some genes that appear to be induced at an earlier stage, not because they are needed there. So the yeast appear to be willing to invest in their future. They are willing to pay a price now because they believe that something good would come out of that later on. How does that confer with the general theme of this talk of doing something in yeast and then asking whether it applies outside in nature? I don't have the time to go through many such examples, but since we published that work, we and others have thought of many additional cases where cells do appear and indeed do predict ahead of time. So the ability to predict and to prepare is not confined to species that have brains. We see it outside of brain-having organisms, but even within our body, I want to share with you just one hypothesis. It's a crazy hypothesis. Imagine that you go outside and you're exposed to the UV from sunlight. This is dangerous. It could be carcinogenic. And we have a DNA protecting pathway that can respond to DNA damage. But perhaps when the damage occurs, it might be already too late. So just imagine that maybe our skin cells have a capacity to predict ahead of time. What could be the bell that would signal to them, analogous to the bell, that would signal to them that UV damage is likely to occur soon? Well, maybe, for example, an elevation in the temperature. What if an elevation of the temperature was a Pavlovian-like signal that triggers skin cells to induce the DNA repair enzyme just in case, because oftentimes there was a statistical association between the rise in the temperature. I even thought to generate sunscreens that would activate ahead of time the p53 pathway or something that would prepare ahead of time. It doesn't have to be relying on an existing physiological process. But this is just to trigger a discussion about that. Switch gears again, very, very erratically. If this is a failed experiment, I apologize in advance. We care about mating and sex very much, and there's no better place to study mating preferences and the genetics of that than yeast. In particular, since yeast already has a mating system, imagine that, and you all know there is alpha and A, two mating types, and if one, say, alpha needs to choose between two different A's, can it exercise any judgment, any preference? Do yeast have anything that allows them to make a choice? And if they make a choice, is it a choice that we can justify, perhaps by the same means that we humans, for example, make our mating choices? So in Saccharomyces cerevisiae, there is a 2 minutes. There's a very privileged collection of many wild strains. It was given to us by Liti, who is collaborating with us, and Sivan Kaminsky in the lab decided to create a mating system, a massive mating system that is based on barcoding of their genomes. She, like in a huge orgy, she mixes all of them together, and barcodes that recombine between mating individuals allow her, through sequencing, simple Illumina sequencing, to identify who mates with whom. And for the first time, we have been able to generate a very large collection, about 100 by 100 strains, of who likes whom. And it turns out that yeast can exercise mating specificity or mating affinities. Some like others more than they like others. It turns out that they have some logic behind their choice, because if you let them choose who they want, they generate usually a higher fitness population than if you force them or if you do that at random. Here again, this theme of looking outside of yeast paid a benefit for us, because we decided to see if the same happens in humans. So don't worry, we haven't done a massive mating human experiment in the lab, but we do that bioinformatically. We have the genomes of many, many people, and each person has two genomes, one from mother, one from father. And computationally, only computationally this time, we can mix the fathers and mothers of all of us and ask what would have happened if mating in humans were random. For example, what would have happened to homozygosity of recessive traits? What would have happened to the distribution of genetic distances between individuals? How does this distribution change with time, perhaps with changing human culture, etc., etc.? So this is a huge project that is now run in the lab, but it was all motivated by what we did in this. So I will stop here and happy to take questions.
So in the tRNA project, where you were looking for suppressors of deletion of one tRNA, have you ever seen mutations in the loader of amino acids in the synthetases?
back then the project was published in 2013, was done even the 2 years before that. We haven't sequenced the whole genome. We sequenced just the tRNA gene, but we do know that the mutation that we found in the tRNA gene, I can't say was necessary, but it was kind of repetitive in every experiment that we did. It appeared again and again, and it was sufficient in the sense that if we went to the original deletion mutant and just put that mutation, point mutation, the fitness was recovered totally. So it does not exclude
I was
additional
wondering
things.
whether there was an alternative
Yeah.
way of doing it.
Yeah, that could have been right, and in a way we should have done the whole genome sequencing, but with these caveats that I said, it appears to be both necessary and sufficient.
Now we have time for maybe a student question, but I don't see very well if somebody
Wasabi.
doesn't see
I see
Wasabi.
Wasabi.
Yes, so that's definitely.
So connected to that same project, could you link the abundance of the tRNAs to their frequency of being the one mutating?
Interesting. So Wasabi is asking, perhaps the abundance of the tRNA, maybe the one that was deleted or the one that rescues by a point mutation towards the one that was eliminated, is predictive of their ability to participate in this game. We haven't checked that actually. It's a good idea. In that case, the abundance, where we started in the lab, the abundance was high because the providing mutation appeared in 11 copies, and by the way, each time we did the experiment, almost each time one of them was chosen randomly to be mutated. So it appears that abundance of the donor is important, but bioinformatically we haven't looked at that particular aspect.
Now I have quick one last question. Safi, didn't you see hands?
Moti, are you a student? You were a student.
Yeah.
You were a student.
Moti is a student of
Never
life. He never stops learning.
Okay, so that qualifies.
Well, fascinating talk. As ever, I enjoyed it very much. I have a question about your first part about codon optimality.
Uh-huh.
When I think of codon optimality, I think that many people do, I think about translation rate.
Right.
When I think about translation rate, I think about co-translational assembly of complexes. So the slower the rate, that's the way I see it, the better the co-translational assembly. So maybe the distinction that you observe, let's say in the glycolytic pathway versus the TCA pathway, is that the TCA forms a complex, whereas the glycolytic pathway does not.
Interesting.
I mean, I guess that back in 2013 you haven't examined it because there was no data, but now there is data.
That's interesting. So Moti suggests that perhaps codon usage programs are not acting just individually to tune the amount from each gene, but rather to generate coordination among different genes, perhaps to ensure that interacting proteins, maybe members of the same complex are simultaneously coming out. That's interesting. Yeah, we haven't checked that. Another effect of codon usage is on mRNA stability. That's very interesting too. So not only translation, but maybe additional
Well,
properties.
I think
Sorry, because we're trying to keep on time, so we have a long break. Zahi, I love the experiment. Thank you so much for a super exciting talk. And now we have the first brave student speaker, Eyal from Dana Reichmann's lab.
Okay, good morning. Thank you for inviting me to speak here today about my project on identifying redox regulating proteins. And I want to start with a proposition. And the proposition is that cells survive in an oxidative world. And the reason that I say that is cells constantly produce reactive oxidant species and other oxidative molecules in the cell, and they're also constantly exposed to extracellular oxidative forces. And while these molecules are very beneficial for the cells, being crucial for different pathways, the accumulation of which can also lead to harmful events in the cell. And in the end of the day, it's a question about balancing, right? How do cells maintain this redox homeostasis, and what is the adjustment mechanism behind it? Now, this was the main question that began this project for us. And I'm a trained biochemist, so if you ask me what is the adjustment mechanism behind anything, my first go-to response will be proteins. And in the case of oxidation, these proteins are your antioxidant proteins that act as first responders, reducing loss agents or oxidized proteins in the cell, which helps maintain the redox homeostasis. So this project actually began with asking how can we identify more of these redox regulating proteins, and how can we do it in a wide approach that will be very efficient. So in order to find redox regulating proteins, we first need a good method to measure oxidation, and in the lab we use a modified GFP-based probe called raw GFP. Some of you might be familiar with it, but basically it's an oxidation-sensitive GFP probe that we usually couple to a GRX1 antioxidant, which helps facilitate its specificity to glutathione-based oxidation. And the way that it's sensitive to oxidation is that it can emit fluorescence in two different wavelengths, the ratio of which changes based on the oxidation of the environment. And by measuring the intensity of the wavelength and calculating the ratios, we can get a direct measurement of the oxidation levels within the cells. And we can do it for the cytoplasm of the cell, but we can also localize this protein to different compartments. So we use this oxidation probing with our physiological model, which is chronological aging in yeast. And the reason for chronological aging, because it is a very good model for intrinsic oxidation that we were really interested in investigating in the lab. So this project began by the works of a former student, Dr. Mital Radzinski, who developed a high throughput identification method for identifying potential redox switch proteins. And basically what she did, she took a deletion library of yeast, so this is a commercially available knockout library of yeast, and she transformed all of the different knockout strains with the raw GFP plasmid in order to measure the oxidation using flow cytometry, and also sort individual cells based on their oxidation levels to either oxidized or reduced populations. She then continued to sequence the identity of the cell. So the knockout library has a genetic barcode replaced instead of the knockout protein, instead of the knockout gene. And she quantified the strain enrichment in each one of the populations, which led to the identification of 1,000 or so potential redox switch proteins. Basically, the knockouts were either enriched in the oxidized population or the reduced population. She did also a lot of analysis following that, and when I joined onto the project, I asked her the question, what can we investigate further between the relationship of these knockout proteins and oxidation itself? So I drafted a list of a dozen or so proteins from our findings, and these proteins are from different areas in the cell, different functionality, and today I will focus on these two proteins, ASC1 and NDJ1. Now, all of these proteins were enriched, the knockouts were enriched in the oxidized population, and AC1 and NDJ1 were particularly interesting to us because they were both nuclear proteins. Both of them are active during mitosis. AC1 is a spindle elongator, NDJ1 is a spindle pole body and telomerase binding protein, and both of their knockouts were highly enriched in the oxidized population. So I began by doing different stress growth assays to see the sensitivity of these knockouts to different stress conditions, maybe just a matter of the knockout protein rather than specificity to oxidation. And when we did oxidative stress, we saw that both of the NDJ1 and AC1 knockouts did show inhibited growth compared to the wild-type strain. But under other stress conditions, and here you can see reductive stress, we saw no significant difference between the knockouts and the wild-type growth under the same stress condition. So already here we see that these knockout strains that were enriched in oxidation also showcase high sensitivity to oxidative stress. So this was really interesting to us and very nice. And the next thing that I asked, is there actually any difference in the oxidation topology in the temporal and spatial oxidation events within these cells? So in order to measure the redox generation in the cell, I went back to our raw GFP plasmid, but this time I used 4 different variants localized to the cytoplasm, mitochondria, nucleus, and peroxisome. I transformed the knockout strains with this plasmid, and I grew the yeast to different chronological aging time points. And again, this is because chronological aging is a physiological model for intrinsic oxidation. So we worked with early log at 5 hours, onset of stationary of the yeast life cycle at 24, and early stationary at 48. I then continued measuring the oxidation levels in each one of the compartments at each given time point, and I mapped the results in order to generate a kinetics map in order to look at how does oxidation generate in the cell and how does it moves with time and space. So here you can see the graph itself, the kinetics map here, and on this radar plot you can see the degree of oxidation at each given compartment at a specific time point. The blue dots are the knockout strain, the red dots are the wild type, and all of the measurements were normalized against wild type, so it's not the degree of oxidation within the AC1 knockout strain, it's the ratio of oxidation AC1 to wild type. And what you can see here was very interesting to us. So at the early log life phase of the yeast, we see a generation of oxidation taking place in the nucleus. So yes, the cell is more oxidized than wild type, we already knew that, but we see a specific hotspot of oxidation event taking place in the nucleus. Now, this was very interesting, but also very logical, because AC1 is a nuclear protein. We saw that the knockout is highly oxidized, and the oxidation does actually take place in the nucleus, where AC1 is missing. Now, when we move forward to the on-state stationary, we see a shift in the pattern. From a single hotspot event, we see a more spread out globalized oxidation of the entire cell. And this globalized pattern also continues into the early stationary. So we can see how the oxidation shifts with time and space from a single compartment in the cell to other compartments, indicating that there might be some sort of communication between the nucleus and other organelles. Now, when we look at the result of NDJ1, we see something very different and very surprising. So NDJ1, I remind you again, is another mitotic protein localized to the nucleus, very similar to SC1 in nature. But we see that at early log, the oxidation event actually takes place in the peroxisome. So again, you have a hotspot of oxidation, a higher oxidation area in the cell. But instead of the nucleus, it's actually the peroxisome, which for us was very surprising. As far as we could find, and as far as we know, the function and locality of NDJ1 has never been connected to the peroxisome. We don't know of any direct connection between the protein and the peroxisome itself, but we see that in the knockout variant, the oxidation generates in the peroxisome. However, we once again see a pattern shift at the onset of stationary. From the peroxisome, oxidation moves to the nucleus. And for us, this suggests that there might be some sort of crosstalk between the organelles, or some sort of oxidation-related or oxidation-based pathways that moves the oxidation stress from one area of the cell to another. And then by early stationary, we once again see this emergence of a globalized oxidation pattern, albeit here both the nucleus and the mitochondria are the leading contributors, but we see how oxidation starts in a particular point in the cell and moves forward into the rest of the cell. So I did this experiment for all of the dozen or so proteins that, all of the dozen or so knockouts from 1 minute, no problem. And I'll summarize it in two key points. So the first point is that there's no direct correlation between the generation of oxidation in these knockouts and the functionality and locality of the original wild-type proteins. So about half of these strains, so marked here in circles, had the oxidation generation in the knockout generated at a different spot than the actual function of the protein. And also the second point is that all of these strains eventually collapse towards this globalized oxidation pattern. So oxidation in the entirety of the cell did appear, but only at a further time point. So just to conclude my brief talk, we developed a high-throughput method to identify new redox switch proteins. We also put it on a database online for everybody to use. I hope I was able to show you here that oxidation probing has the potential to be used to understand cellular pathways and events, and how we can get insights towards potential crosstalks within the cell using oxidation probing. And lastly, cellular oxidation is heterogeneous. For me, it was thought that oxidation is more homogenic, but here I showed you that cellular oxidation can appear in different patterns, in different generations within the cell at different stages, and I hope I was able to convince you that there's much more to oxidation than it appears on the surface. Thank you for listening to me, and thank you for Dana for the help with this project and all of the formal elaboration.
Thank you, Eyal, for a great talk. We have time for one question, maybe from the trainees. I do want to say that we want to have a lot of time for breaks, so we're keeping everything short during the sessions, but I really encourage people to afterwards come and talk to Eyal and talk to the other speakers to continue discussions about the project. So Eyal, I'm going to need your help. Is there any trainee raising their hand?
Over there
but
Yes.
I suspect it's another student of life.
Okay, so did you say about chronological aging here? Did you look at cells which were aging like from in the weeks period, like week, 2-week age cells?
So I didn't show the graph here, but the reason that we focused on the early life stages of yeast is because with yeast, at least with Saccharomyces cerevisiae and all of the wild type strains, you see an increase elevation of oxidation at the early life cycle, at the early life stages before other pathways and other events take place. So it helps to isolate the oxidation-based events. If we look further down the line, a week or 2 weeks, you already have other molecular events that might act as noise or might be hard to discern between that and oxidation.
So the next student that will be submitted to the experiment of stress to show you all your data in 8 minutes is Subham from Moti Hodel's lab. Please, Subham.
So, good morning everyone. My name is Shubham. I'm a PhD student in Professor Moti Khoda's lab in the Technion. And I would like to tell you about my PhD findings, where we identified a novel chaperone function of HSP70. We saw that it binds nascent RNA co-transcriptionally and prevents mRNA aggregation during stress. But before we come to this specific topic, I would like to tell you what our lab does. Our lab studies gene expression regulation and how all stages communicate with each other. We specifically showed that the role of the transcription apparatus is not restricted to the nucleus, but in fact it continues to remotely influence the fate of mRNA in the cytoplasm. And the mechanism behind this, in this example, is that two proteins, RPD4 and 7, form a dimer and bind nascent RNAs co-transcriptionally, and they remain bound to this mRNA in the cytoplasm. And as you can see, almost all stages in the life cycle of mRNA depend upon RPD4 and 7 dimer, which bound to it co-transcriptionally. So we call this process mRNA imprinting. And Shira, the previous PhD student in the lab, asked whether mRNA imprinting is a general mechanism of gene expression regulation. So she developed a high throughput technique called PROFIT to identify proteins that bind nascent RNAs co-transcriptionally. And indeed, she saw a lot of interesting proteins that do this. But when she exposed yeast cells to heat shock, the mRNA imprinting profile changed dramatically. We got interested in two proteins, SSA1 and 2, which are HSP70 variants in yeast. HSP70 offers several pillars of proteostasis, such as protein folding, preventing protein aggregation, regulating degradation of proteins, and even sequestering proteins. But why is HSP70 a chaperone of proteins getting involved with mRNA, which is not a protein? Before we try to understand that question, let's first understand what is heat shock and the heat shock response. During heat shock, cells rapidly repress the transcription of housekeeping genes, and the existing pool of housekeeping mRNAs is sequestered into insoluble aggregates. In contrast, and simultaneously, it activates the transcription of stress-related genes, and stress-related mRNAs are rapidly exported to the cytoplasm. One more time, in the cytoplasm, housekeeping mRNAs are aggregated into translationally inactive aggregates, whereas stress-related mRNAs are maintained soluble so that they can be translated and the cell can adapt to stress. A main question in the field is, how do stress-related mRNAs escape sequestration? So, we hypothesized that given the known function of SSA proteins in regulating protein RNA aggregation during stress, maybe SSA imprints some mRNAs to selectively evict them out of translationally inactive aggregates. To begin understanding this, we wanted a nice strategy to work with only that pool of SSA which is involved in mRNA imprinting. So we used a proximity labeling based approach using BioID and focused on transcription, which is an important step in mRNA imprinting. Here you can see we tagged RNA pol2 with BRA, which is a biotin ligase. So whenever SSA1 and 2 molecules are in close proximity to pol2, they will be irreversibly biotin annihilated. And this biotin tag helps us to work with only that probably small pool of SSA which is involved in a special function like mRNA imprinting. Here you can see under both room temperature and heat shock conditions, SSA gets biotin annihilated, and this biotin annihilation is highly sensitive to labeling time. With a nice system in our hands, we then asked what are the mRNAs that are imprinted. So we grew cells and pulsed them with biotin for a very short time and purified biotin annihilated SSA1 and 2. And then we sequenced the RNA that is bound to biotin annihilated SSA. For this we used a method called crack, which is an advanced version of iClip. We saw that both SSA1 and 2 proteins imprint mRNAs under both room temperature and heat shock conditions. But it imprints a lot more mRNAs specifically during heat shock. We also saw that both proteins have a lot of mRNA imprinting targets in common. In fact, practically all of them are shared between these two proteins during heat shock. And what are these mRNAs and what do they encode for? So we saw that a majority of mRNAs encode proteins that are required for cells to cope with stress, which is expected because we are working on HSP70. But we also saw that many mRNAs encode ribosomal protein components. So we focused on these two groups of mRNAs and then asked what is the solubility of these mRNAs in yeast. So we took the cell lysate and fractionated it into soluble and insoluble portions using centrifugation, and in each portion we detected mRNAs using RT-qPCR. Now let's look at ribosomal protein mRNA. We are looking at the solubility of mRNAs. So the taller a bar is, it means the solubility is high. We see that compared to room temperature, during heat shock, the solubility of ribosomal protein mRNAs reduces, which is consistent with literature that housekeeping mRNAs are made insoluble during stress, such as heat shock. When we look at heat shock protein mRNA, we see something different. We see that during heat shock, these mRNAs are maintained soluble, which is also consistent with what we know about them, that stress-related mRNAs need to be soluble so that they can be translated. But we are looking at the bulk mRNA population, which is a mixture of both imprinted molecules and non-imprinted molecules. So we asked what happens to the solubility of only imprinted molecules. For that, we slightly modified our assay, but this time, from both soluble and insoluble portions, we purified biotin annihilated SSA and the mRNA that is bound to it. And then we measured the solubility of this mRNA. So, when we focus on ribosomal protein mRNA, we see that during heat shock, the solubility of these mRNAs increases. But the reason why we do not see any bars over here is because we couldn't detect anything in the pellet, which means that the solubility is practically infinite. This is the bulk mRNA solubility that we saw earlier, so we can compare it side by side. When we focus on heat shock protein mRNA, one more time we see that during heat shock, their solubility increases. In fact, the solubility of imprinted mRNA overall is much higher than the solubility of bulk mRNA. Here you can see 7 versus 51. So we know SSA1 and 2 imprint mRNAs and evict them from aggregates. But is this really important for cells to cope with heat shock? For that, we decided to disrupt the mRNA imprinting function of SSA. And we did that by selectively evicting these molecules from the nucleus. And for that, we used a strong nuclear export signal. And then we asked whether cells which lack nuclear SSA grow any different compared to wild type cells. So, we grew cells under several temperatures, starting from as low as 12 degrees, around optimal, and then higher temperatures. Here first, we see that cells which lack SSA1 and 2 altogether have a problem in proliferating at 36 degrees Celsius, whereas cells which have SSA but it's not in the nucleus, shown by gray curves, do not have a problem in proliferation. Cells which lack nuclear SSA have a problem in proliferating at much higher temperatures like 38 and 40 degrees Celsius. But this was done under constant stress conditions, which is probably not very relevant in nature, because stress conditions always keep on changing and temperatures might fluctuate all the time. So we one more time took constant temperatures 12 and 36 degrees Celsius, where we already saw nuclear SSA is not required, but we fluctuated these temperatures. So we went from 12 to 36 back to 12, and we repeated this cycle. And now we see that under fluctuating temperatures, nuclear SSA is very much important for proliferation. And as a student, what I have learned from this is, in the past, maybe we have missed out on a lot of important insights by focusing only on constant stress conditions. Maybe now we should start thinking about what happens under fluctuating stress conditions, which I think is more relevant to study stress response. To summarize, SSA1 and 2 imprint some mRNAs, and during stress in the cytoplasm, when proteins and mRNAs aggregate into translationally inactive foci, only imprinted mRNA molecules are privileged to be solubilized from these by SSA1 and 2. When they are made soluble, they can be rapidly translated and cells can adapt and survive stress. This makes us propose a fifth pillar of cellular proteostasis offered by HSP70, which is to control mRNA solubility during stress. And with this, I would like to thank you all for listening.
Subha, we have time for one question. Subha, you will have to tell me who's raising their hand. So I questions?
I just wonder if you know any idea how does the SSA chaperone bind actually the RNA? Is there a motif, binding motif on the...
So regarding the binding motive, we know that some mRNAs have a lot of deposition of SSA on either the 5' end. Some mRNAs have a lot of binding of SSA at the 3' end. And we are now trying to identify if there are any motifs in the mRNA to which SSA has a preference.
Two different regions on the SSA can bind RNA?
No, two different regions on the mRNA
On the mRNA can bind.
So stress-related mRNAs mainly bind SSA through their 3' end, for example.
Thank you,
Thank you very
Suba,
much,
for a
Suba.
wonderful talk.
Thank you.
And now we have a talk from Adi from Shai Ben Aroya's lab.
בבקשה, עדי.
Hey everyone, my name is Adi Akira from Professor Shai Ben Aroya's lab. First, I want to thank the organizers for this opportunity to present my research, in which I focus on understanding whether ADAR editing efficiency is affected by the transcript levels of the substrate. So what is the ADAR enzyme? Its full name is adenosine deaminase acting on RNA, and what does it role in the cell? Our DNA contain non-coding regions with repetitive elements that at the RNA level can fall back and create a double-stranded RNA that can be recognized by the cell as a viral RNA and therefore to create an immune response. In this case, the role of ADAR is to edit this double-stranded RNA by that ion-binded structure and prevent this immune response. ADAR plays another role in the editing of coding sites, a process that is essential for the proper function. What is the mechanism? ADAR editing is facilitated by the deamination of adenosine to inosine, which is read during the translation as a guanosine. So shortly, ADAR edits A to G, and this editing may lead to recording events, for example, the conversion of a stop codon to tryptophan. So many studies utilize this ability to correct a target mutation of their interest. And how exactly they do it? By designing a guide RNA that can bind to the target and mimics the natural double-stranded RNA that recruits ADAR. The problem is that this target is not natural ADAR target, and therefore not all of the transcripts are edited, which can lead to a partially functional protein, and it's not always enough to cure a disease. Therefore, many studies try to optimize the process, for example, by manipulating the guide RNA size or the mismatches within it, because other natural targets contain mismatches. In this study, we want to understand whether other editing efficiency is also affected by another factor, which is the transcript levels of the substrate. To test it, we use the yeast as a research model. Unlike metazoans, the yeast doesn't express ADAR naturally. However, we know from earlier studies that ADAR edits within it when expressed exogenously. And in addition, we can use the yeast as a neutral platform to test editing. Therefore, we transformed our strain with the human ADAR plasmid when ADAR is induced under the GAL1 chromotor. For this study, I created two types of yeast strains in which I inserted a DNA sequence, each time upstream to different gene in the same reading frame. And this sequence created the RNA level a double-stranded RNA that can recruit the human ADAR. But what is the difference between those two strains? In one strain, the gene is expressed under its natural promoter, and in the other, the same gene is expressed under a robust promoter in order to increase the expression level of the gene. That way we can compare the editing percentage as a function of transcript levels. So for all of the strains that I created, I extracted RNA, converted it to cDNA, amplified other target region, and sent it to next generation sequencing to test what is the editing percentage. What we found is that for most of the genes, the editing under the natural promoter expression was higher compared to the robust promoter expression, meaning that probably ADAR edits less when it has many transcripts to edit. So we assume that the reason is that ADAR is a limiting factor, and we wanted to test what happens if we add more ADAR to the cell. To this end, I took the strains with the robust or the natural promoter expression and transformed them with another human ADAR plasmid with a different selection. Then we compared the editing percentage in the presence of 1 versus 2 ADAR plasmids. If you remember, the editing under the robust promoter expression was lower. However, when we added more ADAR to the cell, we increased this editing efficiency, meaning that indeed ADAR is a limiting factor. At the final stage, we wanted to test whether what we found in yeast is also physiologically happening in human cells. To test it, our collaborators Rivka Buchni-Cohen from Professor Erz Lebanon Lab, together with Professor Eli Eisenberg from Tel Aviv University, did a bioinformatic analysis using the GTEx database, a database that contains transcriptomes of different individuals from different tissues. And specifically, what Rivka did was to focus each time on a different tissue and each time on a different gene that is expressed differently in different individuals. And what they found is that the substrate levels for most of the cases affect negatively on the editing percent, and other levels for most of the cases affect positively on the editing percent, meaning that our yeast model reflects physiological conditions. Based on this result, we can predict that a less expressed target mutation is more likely to undergo effective therapeutic RNA editing compared to other mutations associated with the same disease. Therefore, we can increase our chances to cure this disease by focusing thank you all for listening, and many thanks to my lab members and my mentor Shai for their very great support.
Thank you for a beautiful talk and for keeping on time. Really amazing. Yes, we're open for a question.
I have a general comment which is relevant to your talk. Promoters indeed function in transcription initiation, but as we and other investigators found, promoters have much larger effect. They can affect on all stages of the mRNA life cycle, partly via mRNA printing that Shubham has talked about. So when you compare two promoters, you don't compare only the level of the RNA, but you compare the promoter. For example, some promoters can recruit better ADAR than others, and ADAR can function co-transcriptionally, namely, and this is consistent with your observation that when you increase the ADAR level, you see better. So maybe by increasing the level of ADAR, surplus of ADAR find promoters which are not optimal for them. So this is just a general, and you can look at your data and try to interpret them from this point of view.
Okay.
Moti, other is not recruited to the promoter, it is recruited to the
How
double
do you
standard.
know?
This is what is
This
known
is
in the
what is
field.
commonly
Okay.
expected. But many, I mean, the promoter can serve as very good meeting place for many things.
Yes, I
Okay.
love this discussion. I want you to continue it during the coffee break. Definitely include Vadim in the discussion. I want to thank you again for your talk. And our last student speaker just for this session is Soha from Lab Lab.
Hi everyone, I'm Soa, a British student from Elabic Lab, and my research is focusing on how oxidative stress distributes the cellular proteostasis with a particular emphasis on the RAB1 modification pathway.
Can you speak a little louder, please?
Yes. It's okay. As we know that it's well established that during the yeast culture growth, and with the depletion of the glucose in the concentrations, the activation of the mitochondrial respiration, with a result with a high ROS levels that results in oxidation and metabolic and homeostasis stress. And it's expectedly, we saw that wild type cells in the early log phase have a small volume of the mitochondria, and until the late log phase, the volume is more larger and matured and ready to the respiratory activity. And when we observed, when we looked at the morphotype, the mitochondria, the log phase logarithmic cells show a more tubular morphotype, as we see in this figure. And in our lab, we investigate the post-transition modifier, the RAB1, which is a ubiquitin-like protein that uses the methylation enzyme cascade in order to modify the carrying proteins, which in other eukaryotes regulates a turnover of a key cellular targets like cell cycle, stress response, and development. And our data show that there is a correlation between the maturation of the mitochondria and the RAB1 availability to modify the kines under stress. And we know that the methylation pathway is restricted at the deoxyribonucleic shift by multiple levels. The first one is the YH1 enzyme, the first enzyme in the cascade. that we show that it's redox sensitive. And the second is the dimeric E1 subunits, ULA1 and ULA3, that show to be reversibly oxidized. And I will talk about them today. And using a native gel, the ULA1 and the UBA3 observed less competent to complex assembly at the deoxic shift compared to the logarithmic. And in other experiments, these proteins, the ULA1 and UBA3, showed to be more aggregated, not due to oxidation as shown using DGT and TCP, but because it's form hydrophobic detergent assemblies that is sensitive to SDS and but not to NP40. And we continue to investigate the UBA3 protein because it shows more sensitivity to oxidation compared to the ULA1, which is more soluble. And for this, we want to evaluate the aggregates of the UBA3, and for this we used a confocal microscope, and we imaged the cells under the oxidation stress, starvation induced and metabolic oxidation stress, and the increase in the number of the vacuoles in the was a marker for the stress we induced. And in our analysis, using the IMARIS program, we measured the GFP intensity in the same number of areas inside the vacuoles and outside in the cytoplasm, in untreated cells and in the treated cells. And then the mean of these areas was calculated and plotted as the ratio between the vacuolar GFP to the cytosolic GFP. And the results show that upon the three stress conditions, the UBA3 showed to be localized inside the vacuoles. And from here we asked how these facts affect the RAB1 function. And back to Kalins, it's known that in yeasts, the Kalins methylation status doesn't affect its substrate abundance. And we saw that in mutants of RAB1 cycling, the Kalins methylation status is affected, but the expression of the SIC1, which is the substrate of the kalin, and its receptor CTC4 doesn't affected and remains stable compared to the wild type at the post-deoxy and the logarithmic phase. All these arises a key questions. The first one is what is the biological significance for inhibiting the RAB1 system, and where does RAB1 go under stress, and where if this process is linked to the mitochondrial stress. And since that oxidative stress is a marker for a deoxic shift, we test the requirements of RAB1 in acute stress response using a high concentration of hydrogen peroxide for 1 hour, and then the culture used if the strain delta RAB1, delta YH1, and wild type was plated in YBD in serial dilutions. And the results showed that delta RAB1 strain has the worst survival compared to the wild type and to the delta YH1, which harbor the precursor RAB1, the immature RAB1. And from this we conclude that the RAB1 is essential for cell survival under high levels of ROS. And as I mentioned before, the wild type at the early log phase had an undeveloped mitochondria that in the late log its volume it's been more larger. This pattern was shown by the delta RAB1 and delta YH1 strains, but with more larger volume. And this phenotype that doesn't distinguish between the immature RAB1 that accumulate in the delta YH1 to the total loss of it. And when we looked at the morphotype, we saw that the delta RAB1 strain has the more non-tubular mitochondria compared to the delta YH1 and wild type. And I remind you that this both phenotypes is relative to the mitochondrial maturation. And from all this, we suggest that the RAB1 is a repressor that inhibits the pre-maturation of the mitochondria. And for summary, the RAB1 system is shut down under oxidative stress by multiple levels. RAB1 is essential for the survival of the cell survival under metabolic stress, and RAB1 prevents the prematuration of the mitochondria. And big thank for my supervisor lab Peak and my lab mate Van Slim, and a special thank to Shordi Lab for the genomic type ULA 1 and ULA 3, and thank you for your attention.
We can still have any questions? So are you happy? Go ahead.
Any idea what might be the choline targets?
Sorry?
What the targets of the mitochondria? The RAB1 and choline, CRN targets.
Sorry.
What are the substrates?
The substrate.
Substrates
What is the
of
substrate?
RAB1.
Okay. We observed that RAB1 connected to other substrate that not alkaline, and we still investigate this. We don't ensure that it's connected to the mitochondria.
So that's all.